Hello to everybody, and welcome to the 5th ALLODD webinar. I'm honored to present today's webinar speaker to you, Professor Dr. Giulia Rossetti, who has an expertise in modeling key events and diseases, and also drug target interactions using advanced computational methods ranging from molecular dynamic simulations and free energy determination methods to structural bioinformatics and cheminformatics. Professor Rossetti defended 2 doctoral theses in biology at Aachen Technical University and in structural and functional genomics at International School for Advanced Studies in Trieste, Italy. And later, Dr. Rossetti conducted a postdoctoral research at the Institute for Biomedical Research in Barcelona, Spain. And also in 2014, she became a professor of computational molecular medicine at the medical faculty of Aachen University. And she started her research group focusing on multidisciplinary drug design, combining computational biophysics, molecular medicine, and also artificial intelligence. Today, Professor Rossetti will discuss with us part of her research connected to the allosteric modulation in GPCRs oligomers. Thank you, Professor Rossetti, and welcome.
Thanks a lot, Varbina, for this very nice introduction. So I will start sharing my slides. Just let me know if you can see this. So you can see my screen, right?
Yes, we see it.
Okay, and now we can also start sharing. Okay, you can see this, right? Okay, so good afternoon to everybody. I'm very happy to share our research activity with you today. And as I was saying to some of you a few minutes ago, please feel free to interrupt me. It's really meant to be a base for discussion, and I hope that I will be able to be sufficiently clear with you in explaining which are the challenges in this field when you deal with allosteric modulation of GPCRs oligomers. So just for starting, I'm speaking from Aachen, that is this fantastic city that you see here, and this very multi-tower building is the university hospital where my professorship is. Each of these towers represents a different department, and it's very curious because each of these has a different elevator. You cannot really go from one tower to another. It's really challenging. It's like a labyrinth. But my position is also here in Forschungszentrum, but what you can see is like a very nice research center in the middle of a forest, and this is the building where we usually work. So the focus of my research activity is actually biophysical and data science approach towards central nervous system translational medicine. And just to give you an idea, there are several levels at which you can look at diseases in the brain, from the behavioral aspect of the patient to the system to the connectivity. We look at diseases at molecular level. And this because even if drugs are prescribed and taken at the organism level, they always exert their function interacting with a target at molecular level. So you need the molecular level understanding. And when you deal with brain diseases, unfortunately, you have to deal with the neuronal type of cell. In other words, the challenges that we encounter is that we need to always understand which is the impact of that disease on the synaptic transmission. That is a very finely regulated mechanism of neuronal communication. And sometimes you will see this process as just described as a depolarization event, but actually is a very molecular process and is due to the interaction of several proteins that and neurotransmitters at molecular level. So we are talking about not only G protein scaffold receptor, we are talking about ion channels, we are talking about enzymes. So you always need to keep this overall picture in your mind when you deal with molecular medicine applied in the brain. And so the approaches that we follow are either physics-based or chemoinformatic-based, and recently also artificial intelligence-based. And we do not only molecular models, but also cellular models, exactly for the reason that I just told you, that each target is involved, is embedded in a cellular pathway that is relevant for the neuronal transmission. And the perspective is, of course, the development of new drugs. These are our goals, but in particular to go toward precision medicine. And we will discuss this aspect later on. So the focus of today's talk will be on GPCRs. Why GPCRs? I mean, these, despite the fact that are the target of at least 1/3 of the current FDA-approved drugs, they are really at the focus of several brain pathologies. We are talking about encephalitis, we are talking about Parkinson, Alzheimer's, schizophrenia. But the interesting thing is that when you go to look at what has been done for GPCR-based drug discovery, you see that so far in the years, the main focus was mostly based on mimicking or inhibiting the action of endogenous neurotransmitters. And that during the years, there was somehow a failure to expand the number of druggable GPCRs target. There is really a lack of new ligand classes. And there was a key aspect that was always overlooked. And I'm talking about, of course, the allosterism. Now, when I'm saying that the mechanistic understanding of GPCR allostery is behind the study of other families, I'm not saying that we don't know the existence of allosteric modulators. On the contrary, we know a lot of allosteric modulators for GPCRs. And even, I mean, if you take a review like this one of 2018, there are already a lot, there is already a lot of knowledge about allosteric modulator of GPCRs and where they bind. And also in my own institute, we have studies this. We have studies how the energy flow goes from an allosteric modulator to the orthosteric one, how to regulate this network, as well as we show how allosteric regulation of some membrane component like cholesterol can impact on ligand binding. But it's not this I'm talking about. When I'm talking about the fact that we are behind, I'm talking about the realization that allosteric interaction in GPCRs can affect the GPCRs in a manner that they affect signaling. Because GPCRs, as I showed you before, they are the initiator of cellular communication. So when you allosterically modulate a GPCR, you don't just shift the activation or the deactivation or the behavior, of the GPCR in general. You really affect and impact the overall signaling of the cell. And this picture that you see here is already old because it still looks at the GPCRs simply as molecular or vertical molecular regulators. So it still assumes that there is simply a modulator that acts on the GPCRs that is the conduit, and that let's say this initiate a series of let's say intracellular pathway. This is true, but it's very old and is still very, very limited, despite is already complex. And the reason why it is limited is that there is a level of regulation that was so far ignored, that is what we call lateral allosteric regulation of GPCRs, that open up a new entire dimension of signaling. So GPCRs, they are able to interact with several other proteins at the level of the membrane, not only by itself, with itself creating homodimer or heterodimers, but they can also interact with other type of proteins. So in other words, we have an horizontal molecular regulation that so far was simply ignored or not considered. So the reality is that GPCRs are promiscuous objects in the sense that they really like to oligomerize with several other membrane proteins. And this creates a lot of problems from a drug design point of view, because, for instance, if I need to design something against Parkinson, usually I want to target this oligomers that is constituted by adenosine and dopamine, but I should be careful, because if I design something for dopamine, then dopamine, if oligomerized with itself, then it becomes a target for schizophrenia, and so far and so on. So you can see that targeting or given GPCRs is now a little bit, is still relevant, but it is dangerous, because you might end up in several off-target pathways, and actually you might, let's say, really miss the real target that you want to catch. So just to simplify a little bit this complex overview, let's say, for instance, focus on one GPCR. Let's, for instance, focus on dopamine D2 receptors. So the first challenges that I have to face is the understanding of what is called receptor mosaics. So I need to understand which are the possible oligomers that my D2 receptor can do, that my GPCRs can face. But the second aspect, and this is not only true for the dopamine D2, this is true, I mean, I'm taking this as an example, but of course, what I'm saying is also true for other GPCRs. I need also to consider the so-called receptor state, because unfortunately, the oligomeric state and the receptor state are connected. For instance, it is known for the D2 that when it is in a basal state or it is bound to an antagonist, namely when it is inactivated, the majority of the conformational ensemble of the receptor in the membrane is constituted by monomeric form, while there is a very small percentage of dimeric form. This changes if the receptor binds to an agonist, because at this point the equilibrium shifts in part toward the formation of a receptor dimer. And it might be that, I mean, we are considering here homodimer, but it might be again that it shifts depending on the type of agonist to other type of receptor mosaics. So you see, depending on the ligand that binds, this, I mean, there is a different oligomerization state, and on the contrary, different oligomerization state can favor or disfavor the binding of a ligand. So if I need to design an agonist, for instance, I need to have present in my mind that half of my receptor will gonna be in a dimeric state. On the other way, if I want to design an antagonist, I should keep in mind that I might mostly have a monomeric state. But on the other way around, if, for instance, I want to inhibit the receptor, maybe I just need to shift the equilibrium toward the multimeric state, and the binding of an antagonist would not be preferred anymore. So all these mechanisms are still not really known, and they, of course, are regulated by allosterism. The third aspect that we are talking about is selectivity. And this is the third challenge, of course, but this challenge is not really, as you know, peculiar of GPCR. This is a problem that we have in drug design for every family class that is composed by very similar members. And this is the case also for GPCR, of course. But if you add this to level of complexity, then this makes really the reaching of the selectivity very hard. So what we ask to ourselves in my lab, and what we are trying to do in under several, let's say, different project lines is, first of all, can we blindly and fastly predict the GPCR's oligomerization state? And this question seems easy, but it's not, because it's very, let's say, easy to reproduce a dimer that is already known. But it's not easy to have a method that fastly and in an unbiased way predict all the possible oligomerization state of GPCRs. The second is, can we catch allosteric mechanism of state-dependent oligomerization? So can we really understand what is the interplay between agonist, antagonist, oligomerization state, and how it is the allosteric regulation that goes from the lateral, let's say, lateral allosteric mechanism up to the orthosteric side. And the third aspect is, of course, can we exploit this knowledge of receptor mosaic and receptor state related to oligomerization to selectivity? Can this knowledge help us in somehow find a solution for the problem of selectivity in GPCRs? So I will mostly talk about this first two aspects. And in particular, I will mostly talk about what we are doing to answer this question. So the first aspect, can we blinding and fast predict GPCR oligomerization states? Well, in our group, as well as in other group, we follow the approach of using a Martini simulation. So we, in our institute, we have, we have developed a Cosgrain MD platform that it is a web server that helps for the preparation, run, and analysis of Cosgrain molecular dynamic simulation. There is not only Martini, there is also Syrah, and as a Cosgrain approach. And it is simply a portal where you are taken by hand and step by step guided toward, let's say, the construction of a Martini system. And to this platform, what we are doing, in progress, of course, we are we wanted to start to implement also workflow that we have just developed in our lab to predict by combining Cosgrain simulation and free energy enhanced sampling approaches, oligomers prediction. So we are doing this by using two different, let's say, enhanced sampling approach. One is metadynamics, that is the most known, one of the most known approaches for doing this. Also our chair Zoe here has used metadynamics and Martini to reproduce membrane proteins oligomers. And the second approach is a new method called OPES that was developed recently in the lab of Michele Parinello, that is a little bit different from metadynamics. It is called on the fly probability enhanced sampling, and it is nice because it adjusts the bias depending on how far you are from, let's say, the target process that you want to observe. So what we have so far in our hands, and it is mostly due to the work of one of the students in my lab, is oops, okay, oh, sorry. Is a method that without having, let's say, biased collective, without having, let's say, knowledge of which oligomer the GPCRs is going to make, it is able to predict different GPCRs oligomers by changing, let's say, environmental conditions. So, for instance, for the two oligomers, experimentally, more than one interface has been shown to be used for creating oligomers. So in some paper is the transmembrane helix 6, in some other is the transmembrane helix 4, and we show that the preference of one or the other interface and mostly depends, for instance, on membrane composition. So the protocol that we have can predict different possible oligomer of the same GPCR, and and it is sensitive enough to catch, let's say, shift in the environment that have an impact on oligomerization. The second aspect that we want to catch is the mechanism of state-dependent oligomerization, and this is a project that we started thanks also to ALLODD, and right now we have a protocol that is able to predict also this shift in population in the presence of an agonist. But this problem is not only about, reproducing, let's say, this probability of a dimeric and monomeric form of GPCRs. This is something that we can do by combining Martini and enhanced sampling. We are automatizing for giving, also to the user community the possibility to predict this, and with any GPCRs they want. The problem is really to understand, to catch this connection between the orthosteric site and the allosteric lateral communication that is established in a dimer, in a monomer to monomer connection within a dimer. And the challenges of this, and that we are facing one by one, the first challenge is that we need an approach that allows to characterize and predict target flexibility upon ligand binding, because allosteric communication thrive on flexibility. So you cannot have on enhanced sampling methodologies that does not take into account of this. You cannot have, let's say, to study this, for instance, with simply docking or virtual skinning. So you need to have some approach that is able to characterize this flexibility and is able to allow the protein to adapt to the conformation that the ligand induce while binding. Now, the problem with enhanced sampling approach, the majority of them, is that usually, you need to have a great knowledge of the process that in principle you want to observe if you want to predict it. In other words, most of the methods are really biased on your knowledge. So few years ago, there was a development of a variation of metadynamics that use system-independent collective variables. And this means that it allows you to study a process even if you have no idea about how the process will occur. For instance, if it is on ligand binding, if you don't know at all where the ligand will bind, with this kind of approach, you can somehow blindly sample all the possible binding poses and the energy associated to this in a blind way. And this seems great, but the problem is, of course, that it is very slow converging. So last year we did a modification to this approach, and we were able to catch and describe in a quantitative way the change of energy upon ligand binding on the overall protein without knowing where, let's say, without knowing in advance the process. So this is something that we plan to use here to understand how the druggability of the receptor is modified upon the binding of an agonist or antagonist, and how this impact, of course, on the oligomerization state. The second challenge that we have is actually that we really need to understand the differences between agonists and antagonists and how this can impact on oligomerization state, because this seems to be a very trivial question, but it's not, in the sense that the problem with GPCRs is that in the same binding site, you have molecules that can act as an agonist, molecule that can act as an antagonist, and molecule that can act, for instance, as a partial agonist, inverse agonist, weak partial agonist. So it's very challenging, especially if you want to do drug design, for instance, to really understand, even if you take the inactivated form of a GPCRs and you run or virtual screening, it is really challenging to understand if what you will find is really an agonist or is really an antagonist. And really, it is not always clear how to differentiate between the two. And this for us will be detrimental because we already have, let's say, differences in the binding cavity caused by the oligomerization state of the GPCR. So if you are not able to understand how these differences in the binding site impact on the ligand binding mode as an agonist or as an antagonist, then we will not be able to, let's say, understand the regulation mechanism behind. So we found ourselves in the necessity to go a step behind and say, okay, let's first understand what does it really mean agonist on antagonist. What does it really mean to activate or to inactivate GPCRs? And this brings you to the question of what makes a GPCR agonist on agonist and on antagonist on antagonist. And again, this looks like a trivial question, but it's not from a mechanistic point of view. We decided to give it a try to answer this question with a simple machine learning type of pipeline. So, because at the end, this is one of the most common use of machine learning. So I give to my model input that define what it is occur, and then my model, my classifier should be able to understand if what it is in front of it is really occur or it is not occur. And this is what we want to understand with GPCRs. If there is a way that we can, let's say, have an unbiased mapping of the chemical space that really, and the interaction that really render on agonist and on antagonist. So the idea, of course, is to do this with molecular fingerprint, because this is the way that this is the most common way for properly describe the chemical space of a molecule, at molecular level. And for the few of you that doesn't know what it is a molecular fingerprint, is simply a way to represent in a binary code a molecule. Of course, the example that you see here is extremely simplified. So it just tells you if there is or there is not a chemical function in a molecule. But there are much more sophisticated, let's say, fingerprint that we are currently using, some that are only based on the ligand and some that instead embed in this, let's say, type of 1010 vector, also information related to the way in which a molecule binds to a target. So we decided to build up two different classifier, ligand-based and structure-based classifier, where, the difference is only that here you take only in consideration features that are related to the ligand itself without considering the receptor. And instead, in the second case, you take into consideration features that involved also the way in which the ligand interact with the receptor. So just putting in a simple type of model, for instance, you can see here the example with random forest, but we have used also other let's say type of model. But just for the purpose of the discussion, let's stay with an easy one. So let's for instance just use a random forest model. If you use just a ligand-based approach, what you have is that you have clear classification of what is agonist and what it is antagonist, and the classification, I would say, is almost perfect. Now, for whoever of you is expert of machine learning knows that when you obtain a perfect classification, it means that you have an enormous bias in your data. And this is indeed the case. If you see, this is a confusion matrix. I don't know how many of you let's say, knows confusion matrix, but essentially this is a way to, one of the several ways that you can have to evaluate the quality of your model. So it tells you how many true positive are you have correctly classified, how many true negatives, how many false negative, and how many false positive. And when you have no false positive and no false negative, it means that your data are biased. So it means that if you use this model for classifying an ensemble of agonists and antagonists that are not known to be agonists and antagonists, you will end up in mistake, because they will only because this model will only recognize molecules that are similar to your ensemble of agonists, or that are similar to your ensemble of antagonists, but will not be able to recognize something that is even a little bit slightly different from what you have from your data. So we pass to and this is also confirmed by the fact that if you analyze which are the features that are more relevant for this model, you can clearly see that these features are the ones that define the adenosine type of chemical space. That is actually what define most of the agonists of our ensemble, because we are talking about an adenosine receptor. So essentially, our ensemble of ligand of agonists and antagonists is biased toward, let's say, toward the data that are more, let's say, available to us, that are the ones mimicking the natural, let's say, the natural transmitter of the GPCR, that in this case is adenosine. This problem you will have with all the GPCRs, because as I told you at the beginning, drug design for GPCRs has mostly been focusing in mimicking or doing small variation of the endogenous neurotransmitters. So this means that this type of classification will not help us really in understanding what it is an agonist and what it is an antagonist, because you don't have enough variability for answering this question. Therefore, we pass to a ligand protein type of fingerprint, where you embed information relative to the interaction that the ligand is about to establish. And in this way, we have a much more, let's say, balanced situation. And also the amount of features that are considered are really different and depends on the different type of interactions that agonists and antagonists are able to establish. And with this model, we are able to have a clear distinction of what really means being an agonist and what really means being an antagonist for that particular GPCR that we are examining. We have done like proof of concept type of studies. We have performed a case of virtual screening among, molecules that are commercially available, so nothing really new. But we wanted to see if the molecules that we find from simple virtual screening can be classified, can be filtered out as agonists and antagonists using our model, and if we were able to finally detach ourselves from the adenosine type of scaffold. So if we introduce something different, will our protocol able to recognize agonistic or antagonistic type of interaction? Because this will be relevant on the type of modulation that they will have on the oligomerization state. So we end up with a series of molecules. We'll filter out, and we end up with this potential antagonist that, as you can see, they have nothing or very few in common with the normal scaffold of endogenous ligand for this GPCR, that is the adenosine receptor. Now, can we really say that these are agonists? The real answer is that so far we only have our model, but we really cannot say. And we cannot say because what makes really an agonist or an antagonist is what happens at the level of the cellular pathway. So it is not sufficient to define agonist on agonist and antagonist on antagonist if just they are able to bind to your inactive or active state. You can dig out the interaction, which are the correct interaction. You can select the ligand like we did that are actually going in the direction of having or not having the correct interaction. But these are not still, let's say, 100% sure to be agonist or antagonist because you are not taking into consideration one of the major aspects of being a GPCRs. That means being embedded in a network. So here come in our help the subcellular modeling. So in principle, it has been this, system biologists has been doing this since several years. They can, let's say, mathematically reproduce cellular cascade, and they can, let's say, try to predict what would be the answer of a given cascade, having an input, let's say, in some kinetic parameters. Can we use something like this in our study? So can we connect the molecular aspect of a simulation, of a docking, of a virtual screening, of a molecular type of regulation with the subcellular aspect of mathematical modeling like the one that is done in the system biology community? And this is actually what really distinguishes between traditional drug design that is fixed in one target, one ligand, and something else. So traditional drug design, I love it. You can really achieve high specificity. You can reduce the side effect, but you have a limited understanding of what the target will do within a cellular context. And so you can really not predict adverse off-target effect or adverse toxic effect. On the other way, when you do system pharmacology, you can have a broad biological mapping. You can have an understanding about how a network dynamic respond to a given perturbation that could be a ligand, but you don't have any molecular understanding that you absolutely need if you want to deal with allosteries. So we try to mix these two aspects because especially for GPCR, this is relevant. As I told you, adenosine can form, let's say, dimer with dopamine. So you need to have this molecular understanding, and you need to know how these oligomers can be regulated, how the binding of an agonist on antagonist shifted the equilibrium, and you need to introduce this information in this, let's say, in this concept of system pharmacology. And this is exactly what we have developed in collaboration with Rui and with Alejandro Giorgetti that are, Alejandro is a professor, Rui is a postdoc in our institute. We have developed what we call a structural system biology toolkit that will be, I think, a matter of weeks available as a web server as well. That is a framework to run a system biology simulation. So essentially, it's is able to take in input molecular data like simulation, like dockings, result of virtual screening, use on agonist antagonist discriminator similar to the one that I showed you before, and apply the correct, let's say, biochemical network model. This step is fundamental because if you don't know if you're using an agonist or antagonist, you don't know which biochemical networks model to use. So this step is necessary to understand the correct equation to use. And we did this, and actually, and we applied this on our presumed antagonist. And I can tell you that we were able really to predict with very good, let's say, quality, the trends of our, let's say, binding curve with respect to the one that was actually experimentally measured. So this is the ones that we predict with our approach, and this was the ones that that we, let's say, that our colleagues measured. So this is to show you that if you put together, let's say, the single molecule traditional drug design, where you can put in all the information concerning oligomerization, genetic mutation, molecular interaction, allosteric effect, and you, fuel this in a system pharmacology type of pipeline, you can, let's say, really understand or predict what could be the response of the network. Of course, you cannot do this with whatever system comes into your mind. You can do this only with some GPCRs, or better with the systems that for which you have a lot of information, because for instance, we don't have a system biology equation for all the cellular pathway existing so far. We have the ones for the most studied one. We don't have, we don't have, let's say, enormous amount of data for all antagonists and agonists of all GPCR. We only have for the major classes of GPCRs, and these are relevant for if you want your machine learning model to proceed. But for all this, let's say, subcategories, so for very well-known GPCR and biochemical network, you can, let's say, put together these two aspects and have actually a good response. And this is actually what allows you to go toward what we call precision therapeutics or personalized medicine. And this is indeed what it is at the basis of the current research industry partnership that we have in our institute. So with this, I would like to close here my presentation. As you can see, I have only a sad picture on Zoom because we didn't do yet an in-person retreat. And so this is the last retreat that we did, unfortunately, was let's say, online. So I have only online pictures of the people in my institute. And I acknowledge the Jülich Supercomputing Center and, of course, all the funding agency that support the research in my lab, including ALLODD. And of course, thank you for your attention.
Thank you so much for this presentation. It was so interesting. You did an amazing work.
So thanks a lot. Zoe, should I stop the registration or should I register also the questions?
To record the questions?
Yes, I will stop recording.